presentations about the Cellular Level of Organization unit in A&P 1. This first one's on the cell membrane. The cell membrane is the way that any cell interacts with the environment around it. It's very important in regulating what can get into or out of the cell, or more importantly, keeping things from getting into or out of the cell. First, let's look at the phospholipid. This is a macromolecule that's the majority of what makes up the cell membrane. It's essentially based off of the triglyceride. There's a glycerol molecule in it which has attached to it still two hydrocarbon chains. One of them should be unsaturated, as we see in this picture, with a little kink from a double bond in it. The third fatty acid chain, which would be present in a triglyceride, is no longer present, and in that third position on the glycerol molecule, a phosphate group and a little bit more has been attached, making for an interesting molecule with two important properties. The hydrocarbon chains that are essentially the fatty acids are hydrophobic, being hydrocarbon molecules essentially. And the glycerol and phosphate group, plus what's attached to it, is hydrophilic. This gives the molecule the somewhat unique aspect of having a dual personality, or dual properties, and it makes it possible for this to set up an interface between differing liquid compartments within the cells and between the cell and the extracellular space. Because of the dual properties within a phospholipid, it will naturally take on a configuration in large groups in a water solution, or water environment, I should say, where it forms a phospholipid bilayer. Now, in small numbers, it'll form a different structure called a micelle, but we're more interested in the phospholipid bilayer structure. What happens is the phospho group polar head ends up being exposed to the water, and the hydrophobic tails tuck in and in two layers face each other, so that the core of this bilayer is a hydrophobic region of those tails, protected on either side by the hydrophilic head regions of either layer. One side, if we're thinking about the cell membrane, is going to be towards the extracellular space, and the other side is going to be towards the intracellular space. Phospholipids certainly make up the majority of the cell membrane. Another lipid that we find incorporated in with it is a cholesterol molecule. There are a few of those indicated in this image. Then two other types of macromolecules are found associated with the cell membrane. There are proteins that come in a number of different forms, only some of which are really indicated here, and even then they're somewhat schematic. And then there can be carbohydrate structures related to the cell membrane. Two versions are shown here. There's a glycoprotein, which would be one of the proteins in the membrane, having a glucose-based chain or branch chain attached to it, or a glycolipid, where the lipid structure, essentially the diglyceride portion of a phospholipid, has a branch carbohydrate structure sticking off of it. For the most part, glycoproteins and glycolipids are in the outer leaflet, or the extracellular facing side of the cell membrane. They often work in cell-to-cell interactions, recognition with things like the immune system, or those types of things. They don't tend to be on the intracellular leaflet side. Proteins, however, will often cross over the entire membrane, Some are localized just to one side, possibly more often on the internal side, but that's not exclusive. But there are a number of transmembrane proteins, or sometimes they're referred to as integral membrane proteins, as they're integrated into the membrane. An important part of the membrane is channel proteins, or pores, that are going to make it possible for things to cross through the membrane. As I said earlier, an important function of the cell membrane is regulating what can cross from one side to the other, what can get into or out of the cell. The next two pictures sort of demonstrate that, because there's two different aspects of how that can be regulated. And it has to do with the way that those molecules interact with the cell membrane. Nonpolar molecules, essentially meaning lipids, as long as they're not too large, can cross directly through the cell membrane, mostly unimpeded. And so we see here three different snapshots in time as a concentration of small nonpolar molecules on the extracellular side of a cell membrane will work their way through the membrane until they have diffused to an equilibrium on either side. For molecules that are polar in nature, meaning that they're made of polar bonds, or maybe that they have a charge to them like an ion, such as sodium or potassium or something to that effect, the only way they can cross the membrane is if they have a channel or a pore through which to go. So these integral proteins that can be channels will let small particles through, such as ions. Larger things will often have to be transported through by a specialized protein, what's called a carrier protein here, sometimes referred to as a transport protein, that will change shape and actively, or based on indirect energy expenditure, move something across the membrane. Now, the difference between nonpolar and polar molecules crossing the membrane really has to do with the nature of the phospholipids. The hydrophilic head groups on the outer and inner surface of the membrane are interacting with the water, and are polar, just like the things dissolved in the water. However, the hydrophobic tails through the core of the membrane are existing in a hydrophobic, or water-free, essentially, region. Polar molecules can't move through the hydrophobic core of the membrane. They have to move through a protein. Now, proteins, because of how they're made with amino acids, can be tailored to fit into the protein. The outer surface of the channel, as it's passing through that hydrophobic region, is also going to be hydrophobic. The inner surface of the pore is going to be hydrophilic, where the charged particle can pass through. And then, of course, the extracellular and intracellular edges are also going to be hydrophilic as they interact with the water solutions on the outside or inside of the cell. The carrier or transport proteins are going to be similar in how they interact with their molecules, just with the added feature that they can often change shape as they're physically moving something across the membrane. To understand how these things move across the membrane, the small nonpolar molecules or the polar molecules or charged particles that were referred to in the last couple of frames, you need to understand a little bit about the concept of diffusion. Diffusion is the property of substances such as this solute that we see in a solvent that is going to diffuse to equilibrium. Now, here are 4 snapshots of a dye being added to a water solution in beaker. At the beginning, you see that the dye is rather concentrated right where it's dropped in. As the dye particles run into and interact and move around within the water solution, they'll spread out, and they'll keep doing that until they spread out throughout the entire solution to equilibrium. What's happening on the outside or the inside of the membrane is essentially represented here. In the first time snapshot of the small nonpolar molecules in the earlier slide, they were all on the outside of the membrane. The glass represented in the beaker here, however, is an impermeable membrane. Nothing can get through it, so the dye expands to, if you will, diffuses into the space that is the water up until its barrier. However, a cell membrane is a semi-permeable membrane, and so it's possible for things to move across it. And diffusion will do that. When there's more outside than inside the cell, the solution, the substance will diffuse across a cell membrane. It might take a little time to get across a cell membrane because of the nature of how it's getting through, but it will continue to move across given the chance until it reaches equilibrium. Especially for charged particles like ions, by controlling the openings, the pores of those channels, it's possible to set up concentrations inside or outside the cell that are different. And by opening those channels, it's possible to allow diffusion to drive a passive change in that concentration. This drawing suggests the sort of thing that might happen. Again, we're looking inside of a beaker, but now we're looking at a solution that's divided by a semi-permeable membrane, which is represented by the dashed line here. So imagine the holes in this dashed line represent pores that will allow some things through. Now, what we have on either side of that line right now is a solution. The pink background of that solution, imagine that's the water, and it's made up of water molecules, which are awfully small. And then the gray spheres or dots represented here, imagine those are glucose molecules. So we're looking at sugar solutions here. On the left side of the first beaker, there's just a few solutes there in the solvent. It's a very weak sugar solution. On the other side, on the right side of the first beaker, there's a lot of solutes in the solvent, relatively speaking. And so that would be a concentrated sugar solution. The holes, however, are too small for the solutes to go across. Now, in this situation, the smaller molecules, which would happen to be the water, will move across the membrane. Water diffusing across a membrane is a process called osmosis, which is a special case, but what needs to happen is the concentration of solutions on either side of the membrane needs to be the same. It needs to be an isotonic solution. Isotonic meaning same solution or same concentration. So in the first part of this picture, there's a hypotonic solution, a low concentration solution on the right, and a hypertonic solution, a high concentration solution on the left. With a semi-permeable membrane that will let the water cross but not the solutes, the glucose molecules, what will happen is the water from the hypotonic side will move through osmotic pressure onto the side that's hypertonic. And enough water should move so that the two sides become the same concentration. Now the volumes obviously are very different, but the concentrations are supposed to be the same. These aren't the exact numbers, but imagine that on the right side now in the second beaker, there are 7 glucose molecules in 1 microliter of water. On the left side, there's 28 glucose molecules in 4 microliters of water. So that's the same concentration. There's 4 times as many solutes in 4 times the volume. That's not the exact numbers that are represented here, but it's a pretty good representation, a pretty good idea of how it works. Water will diffuse across a membrane when other solutes can't, so that the solutions on either side will become isotonic. This series of pictures represents a somewhat realistic representation of that idea of the osmosis that I just described. Here we're looking at red blood cells in a solution. So the pictures are supposed to represent what we would see through the microscope, just the field of view of a microscope. And so these are red blood cells that were in a solution. The first slide represents red blood cells that have been added into a hypertonic solution. Now, in the body, with a few minor exceptions, all bodily fluids have the same tonicity, if you will, the same essential concentration. Exactly what's concentrated in them, whether it's sodium or chloride or some proteins or whatever, might vary, but the concentration of stuff in the water will be constant. It'll be an isotonic solution everywhere. If we put cells into a hypertonic solution, which might be something so extreme as red blood cells into seawater, for instance, then the intracellular fluid would be hypotonic compared to the extracellular fluid, which is the hypertonic represented in the label here. And so the water from the cells will move out into the extracellular fluid, causing the cells to shrivel up. It's called crenation sometimes, if you come across that term, but shriveling up is plenty of explanation what's going on there. The center picture represents what cells should look like in a normal situation. So if the red blood cells have been put in plasma, serum, whatever you want to call it, a regular bodily fluid, they should maintain their normal shape. There's no net movement of water in or out of the cell. There is movement of water in and out of the cell, but the movement cancels out. As much comes in as comes out. The third possibility is putting the red blood cells in a hypotonic solution, and an extreme example of that would be if you put the red blood cells into distilled water, so there's nothing dissolved in that. So it's so hypotonic that it's no concentration of anything, it's just pure water. When that happens, the water in the hypotonic extracellular space is going to try to flood into the red blood cell because the intracellular fluid is now hypertonic. And in that very extreme situation of putting the red blood cells into pure water, distilled water, the osmotic movement of water into the cells will be so extreme that they'll blow up like balloons until they burst. And in fact, one's depicted in the picture here as bursting. That bursting of the cells is called lysis. So, and since we're talking about red blood cells, it's specifically hemolysis, lysis of hemo cells, if you will, blood cells. If the hypotonic solution is just a little bit less concentrated than your average bodily fluid, then it would be less dramatic, but the cells would still bloat up a little bit with excess water. And sometimes when people are retaining water when they feel bloated, it's because of a change in the balance of electrolytes or something like that, and the movement of water within the body between different areas can lead to a little bit of bloating, but not to the point of making anything burst. It can be a little uncomfortable, but not horrible. One way to remember the process that we're talking about here, water moving from hypotonic to hypertonic solutions, is the cute little couplet, rhyming couplet, hypo gotta go. So if you're water in a hypotonic solution, you gotta go into the other solution. So it's always going to be water moving from hypotonic to hypertonic, causing this kind of change. There's another type of movement that the cell membrane regulates that we want to consider here, which is when the cell membrane is actually changing and moving things physically across it, not relying on channels or transport proteins like we see for smaller molecules and particles.